Charlotte Deane. I will briefly introduce Charlotte. So she's a professor of structural bioinformatics in the Department of Statistics at the University of Oxford, and she's also the chief scientist of Biologics AI at Exscientia. She was also recently appointed member of the Order of the British Empire in the 2022 birthday honors for services to COVID-19 research. She works on diverse problems across human informatics, protein structure, small molecule drug discovery using statistics AI and computation to generate biological and medical insight, and she always aims to make her tools widely available to the community. And today she's talking about multi-property optimization of antibodies from structure to binding to developability. Thanks so much, Charlotte, for being here today with us.
Thank you, Victor. So good morning, good afternoon and evening. I think I'm in evening, but I suspect there are some people who are still right at the beginning of the day. First, I really want to thank the organisers for inviting me to be part of such an exciting lineup of speakers. I've been really enjoying listening to everything that's been happening. And I went for a very big mouthful of a title. And I think actually Peter might have already given a much better answer to the title that I've given than I'm about to. But I'll attempt to add a little bit more around this area. I'm going to describe some of my published work, but I'm actually going to focus on a couple of things that aren't published yet, that are just about to become available, because it's more exciting for me to be able to talk about new things. And I'm just going to repeat what Victor said in the introduction at this point. All of our tools are freely available and downloadable to everyone. So if you want these tools, come to our website, and for the ones that are unpublished, feel free to email me and we will share them. I'm going to start with something which I think pretty much every speaker has mentioned, which is it doesn't matter how good your algorithms are, it doesn't matter how clever you are at writing very good computational code. If the data is not there, nothing you do will be useful. And in some senses, it's not like you must always have tons and tons of data. That varies by what you're trying to predict and the algorithms you're using. But the data that you have has to be curated in such a way that it's possible to trust it and feed it directly into your methods so that it is comparable and usable. I mean, I think Conrad gave very good explanations of this during his talk. My group started a long time ago on this path, and actually I should say Conrad was quite a large part of the effort when we were doing this. And I'm just going to talk about our three main databases before I kind of jump into the methods, because you'll see that I'm using this data again and again and again as we work through the methods. So the first of our databases is the structural antibody database, and I hope that many of you have heard of this. We built this basically out of the frustration that if you type the word antibody into the PDB, the one thing you didn't get back was the list you really wanted, which was the list of all of the structures of antibodies. SABDAB is a fully automated updating collection of all of the freely publicly available structural antibody data. And you can go and look at it and play with it. It collects that data, it curates it and presents it in a very consistent manner. It also contains nanobodies. There's about 1,000 nanobodies in the database now. Alongside that, we have TheraSABDAB, and this is a self-updating database of immunotherapeutic variable domains. And we also link that directly to SABDAB. So of course, you can see which ones have structural representations, so you can see where you have that information. And we harvest those sequences as they are released by the World Health Organization. There's just over 800 entries there. And the nice thing that has happened with these two databases is sort of the completion of the circle, because now the PDB is using these databases as their annotation of antibodies. So we are now actually getting to the stage where you can actually start using the PDB in the way that we hope to right at the start. The last database is observed antibody space. Oh, promise I didn't touch anything. No idea where that went forward. This has now about 2.4 billion antibody sequences in it. And once again, it's fully downloadable. You can download the entire database if you wish to. And once again, what we've gone for is trying to create that database in such a way that you've got all of the annotation data beside each of these sequences. So the species, the immune state, the individual, if we have it, if there are disease states, age, anything that it's recorded with them. But we've also made sure that it's sorted, cleaned, annotated, translated, and numbered in a very standard way, which is incredibly important. And I think this was something somebody asked about earlier in terms of amounts of sequences. There's about 200,000 paired sequences in there in a kind of non-redundant sense. So you're starting to get an appreciable amount of paired sequence space, but it is still relatively small. So on to the tools. Now, this slide gives a very, very quick overview of, I would say, most, but not all of the tools that we have for predicting properties of antibodies, designing antibodies, but also just annotating the data that we have available. I'm going to go a quick whip round of this slide, and then the ones in red I'll talk about in a little bit more detail in the talk. So if I start from the top left, we have Anarchy, which is our numbering program. So that can take antibody sequences and number them into whichever numbering you would like. And it's very powerful. And I'll show you why it's so useful to us as I talk about the tool that is underneath that, which is something we haven't published yet, but is quite sort of the most newest thing we're just about to drop out from our tool set, which is a way of being able to rapidly and exhaustively search these very, very large databases. I think Lucy was mentioning the challenges once you have these very large numbers of data points, working out computational ways to be able to handle that data is also very important. We also have, and Conrad, I think, mentioned this earlier, AbLang, which is one of many antibody language models that are out there. There are many others, and they're all sort of similar but different to each other. Then we have a set of tools around structure prediction. And in this, I will only talk about AbLooper, which is our kind of deep learning structure prediction tool. And I'll tell you a little bit about how that works. In antibody property prediction, there are many different properties that you might like to be able to predict or control for an antibody. For example, you might like to control how human your antibody sequence is. So I'll talk a little bit about HumAb, our humanization tool. I'll talk a bit about Paragraph, which is something that we've very recently put on the archive, which is a way of being able to accurately predict paratopes very, very quickly using very simple methods so that you can interpret the data very easily. And I will talk a bit about TAP, one of our slightly older tools, which is for this analysis of the developability of an antibody. And there are others there. If you're interested in epitope prediction, you can look at our methods, paratyping, abligity, and space, and full pipelines for thinking about predicting antibody antigen binding. There's also a deep learning method called DLab. So first onto Ka search. Now, this was a kind of a bit like our reason for building SabDab. The reason for building Ka search was we wanted to be able to search our sequence database, but actually it was getting to be very hard work. When we initially released OAS, it had just over 500 million sequences in it back in October 2018. We're now at this sort of 2.4 billion. So you can see this is a very rapid growth in data. And the methods that we had to search this were very slow. And the reasons you want to search it are very obvious. You would like to be able to explore the origin of your sequence. How does it belong with all the other sequences in the data set? Does it relate to those which come from a certain disease? Is it more human or is it more mouse? Does it look like things from natural repertoires? Or even, can I find other things that might bind in a similar way? So I don't want to search the whole sequence, but I just want to find things which have very similar CDRs, or I want to find things with similar paratopes. And so then I can start searching for very specific areas, but perhaps antibodies with different, if you like, other properties, and be able to use that as I go forwards. Ka search is built to be as rapid. It's still not quite as fast as I would like it to be, but very importantly, exhaustive. And I'll show on the next slide why that's so important in terms of thinking about antibodies. What it does is it's built to search the entire database. And like many of these very fast sequence searching tools, the important part of this is actually a preparation part that you put onto your database. So you have to prepare a database before searching. So that's like a pre-calculation. The code for doing the preparation is also available. So if you want to download OAS data in the prepared form or in its normal form and add to it your own data, you can do that and you perform the search in the same way. It's also very flexible. You can search for across the entire antibody sequence, just across the CDRs, just on the paratope residues, whatever you feel would be the most useful piece to search against to see if there are other antibodies within this space that share those properties. So these are kind of the two reasons why we had to build this method. And the best way to do this is to show you the comparison. There are already two antibody-specific methods for searching sequences against antibody databases. The first of these is in iReceptor, where actually they have over 5 billion sequences in that database. So they have an even bigger search problem sitting there than we do in OAS. But the only method they have currently available is searching for an exact CDR3 match. And I can completely understand, because it's put up as a web server, and I wouldn't want to put up a web server searching against 5 billion sequences if I was going to do a more complex search than that. So there's no kind of, this is exactly the way you would think about this. Another of these is AbDiver, which is part of Conrad's work, where you can search against a subset from OAS, a sort of carefully curated subset to try and make this as diverse as possible. And you can search by, if you search by chains, you want to search against the whole of the VH, but you have some conditions here in order to be able to make that search very fast again. And as I said, both are only available as web servers, which for us wasn't very helpful if we wanted to do very large numbers of searches. So I guess my interest is not just, can I search my one sequence of interest against OAS? My actual interest is, can I search hundreds of sequences against that? Now, on the right-hand side is the other reason that this was necessary. Now, there are many, many protein sequence search tools. I am certain people here have probably used BLAST at some point, even if they don't know that they have. And MMSeq is probably the most famous recent tool about being able to be very, very quick, and is often used on the front of AlphaFold predictions because of that. Now, the issue with these tools is they are built to be used in general protein space. General protein sequence space is actually relatively diverse. And so their methodologies, in order to speed up their runtime, rely on, if you like, simple clustering metrics to choose sequences to search against to go as fast as possible. What I'm showing here on this graph, if you look at the blue bars, and the kind of y-axis for the blue bars is the right-hand side of the graph, which is the closest identity identified. If I look at the lighter blue, how often I actually get the right sequence out of a 10 million set of sequences, so find the closest match, if I run BLAST, is only 19 out of 100 times when I tried to do this. For KA search, it's every single time, because it's an exhaustive search. And that's why I need it to be exhaustive, because actually the standard methods are unable to get past this issue of this very different sort of sequence space you're searching in. On the left-hand side axis, which is the orange bars, this is the runtime of searching a sequence against 10 million sequence size database, just to give an idea. And we are not the fastest method there. We are considerably faster than things like CD-HIT and BLAST, but we are slower than MMSeq. But of course, MMSeq is not in any sense, as you can see, it never in its top 100 actually gave us back the closest identity sequence in the antibody database. So it's not going to find you potentially the sequences that you are interested in looking for. Just to give a sort of little idea of the kinds of things you can use this for, I'm giving you one sequence example here. So this is immune repertoire mining. I'm just looking at a particular antibody, COVOX 253. And I'm looking at the number of sequences that are over 90% identical to COVOX 253 that I find in OAS. If I search the whole chain, I get a large number of sequences. And if you look at what I've got on the kind of axis here, I've sort of split it by where the disease of patient comes from. So I find a lot of things which the patient has no disease. And that's unsurprising, because the vast majority of what I'm finding is probably just very strong matches to the framework. If I search across the CDRs, all of a sudden, everything I find comes from one of the sets within the immune repertoires, which is from a SARS-CoV-2 patient. And I can make this more precise. I can look for things just in the paratope. So I can take the paratope residues and I can say, find sequences that match and contain all of those residues. And there's about, I think, somewhere over 100 sequences which have identical sets of residues at the paratope. well, over 90% for that. So you can start to see how you can use this tool and play with it to try and get at different questions and be able to answer different things. Hopefully, I've convinced you that I have a rapid and exhaustive sequence identity search for known antibodies. It is fast, but I will be honest, we are not going to put this up as a web server because it isn't fast enough for that. But one of the things I should say is that though if I put a single sequence in, it takes 20 minutes with 2 CPUs, if I start doing 100 sequences all at once to do the search against, so if I put 100 sequences and want to search them against all of OAS, it scales very, very well. So at that point, it is 10 times faster per sequence. So it takes 2 minutes per sequence. So you can get these very big speed ups to do the large scale searching that I'm interested in. And you can search over user defined regions. You can choose what sets of sequences you want to get and look at the metadata as always. I'm now going to move on to something that we published last year that kind of follows on from all the work that people have talked about around structure prediction. I think the leap forward in structure prediction from AlphaFold 2 made everyone sit up and think, well, if you have that, you can do so much with structure. But one of the main issues with AlphaFold 2 is not its accuracy. It is actually something which Conrad mentioned, it's how slow it is. If you're only interested in predicting the structures of 10 proteins or even 100 proteins, the fact that you might take 20 to 30 minutes to make a prediction is fine. If I'm interested in being able to predict very large amounts of this antibody sequence space, then I will need to be able to predict much, much faster. And the rates that we're talking about is thinking about CDRs for 100 structures in under 5 seconds. And if you want to perform a full minimization to ensure there are no problems within that, it's about 30 seconds, a bit less, sort of 20 seconds per structure. So this is still an enormous amount faster. And in terms of accuracy, I've just put the CDR-H3 accuracy on the Rosetta antibody set there. And I think it's good to say here, the star there for AlphaFold 2 is because some of those antibodies could be in the training data for AlphaFold 2. So just to be totally accurate around that. The numbers here always move around a bit depending on what you test on. So I don't like to say that we're the best. We've compared to, for example, IgFold, which is one of the more recent methods that come out for antibody structure prediction. And what you see is a lot of these methods are operating around the same numbers. So I do not necessarily think that AbLooper is better than every other one of the deep learning methods that come out specifically for antibodies, but it is also not worse. The other property that we were very keen for AbLooper to have is shown on the graphs on the right here. And I'm showing you AlphaFold 2's confidence score across the so sort of for the CDR-H3. And what you can see is actually it can't really tell you if the CDR-H3 is any good. So even the very good CDR-H3s with low RMSDs, the confidence score basically is flatlining for you. So if you made any kind of cutoff, you start losing good stuff as well as bad stuff very, very quickly indeed. And you keep a lot of bad stuff. Whereas with AbLooper, you can see our confidence score versus our CDR-H3 RMSD. It's not a perfect correlation, but you can see how it's perfectly possible to draw a vertical line on that plot where almost everything you remove is bad, and you leave everything that's a good prediction, but of course some bad predictions still remain. I think that this type of understanding of the confidence of the models is also going to become more and more important, particularly because we are continuing to start thinking, well, we'll build these models, and people talk about with AlphaFold, and then we'll use them for the next part of the program. But if we do not understand the errors in those models, we are just putting errors in to propagate. So hopefully people will continue to work on and think about, and I think a big issue, understanding how we create really good confidence scores for all our different types of machine learning models and other types of models that are being built now. Next, the therapeutic antibody profiler. This we developed some time ago, and it's just a set of developability guidelines, thinking about things like aggregation, flexibility, topology, viscosity, and pore expression. And it really, I'm not going to say very much about this at all now because I do not have that long in the talk, but I would encourage you to go and play with it if you're interested in this, because it is also a display technique of being able to see where your antibody, where your sequence of interest sits with respect to what is expected for these properties. It is less of a kind of nice clear cut, this is definitely bad, but more an understanding of how you fit into the general space of what antibodies are like in terms of being able to be good for not aggregating or expressing well. And then a tiny bit, I feel like I'm racing, but it's a short talk and I want to talk about one new tool, and second new tool as well, is a little bit about HUMAB. HUMAB is a machine learning approach for this problem of antibody humanness. The majority of antibody therapeutics still come from non-human sources. And so in some way, they must be, in inverted commas, made human. And that is made safe enough to put into a human, they don't raise a serious immunogenic reaction, because that can be very, very bad for a patient indeed. And this is a really important property because it's a very hard one to test. And what I mean by very hard is fairly obvious, which is I'm not allowed to go around injecting antibodies into people to find out what will happen. So it's very important to have as good a prediction of immunogenicity before we get to serious experiments and, onwards as we can, because there really, there are quite good, very complicated experimental techniques to get an idea. But actually, the final fundamental test for this is patients who have anti-drug antibodies. What we built here used OAS. So that vast database of sequences, the vast majority of it is human. So you are able to build a model where you can say, tell me if this is human or not human. And that's all we trained it to do. We just trained it to separate into two categories. And if you look at the table on the bottom left of this slide, you can see the ROC AUC and the YJS statistic. Basically, the closer to 1 that number, the better. If it's 1, it's perfect precision of being able to do that separation. And this is on a totally separated test set. And there's another method there, an LSTM, that was published around slightly earlier than HUMAB, just for comparison. And what you can see is the number is nearly always 1. And the reason behind this is actually it's not that hard a question to tell whether a sequence is human or not once you have these very large data sets to train from. What was much more exciting and interesting to us is the picture on the bottom right. This is a graph where we've got data with a set of antibodies about the percentage of patients who had ADAs, anti-drug antibodies. And all we've done is we've stratified our data into negative scores, so that's the far right column, positive scores, so we would have said this was a human sequence, and high positive scores, just to see if it really matters. Yep. And what you can see is if you are getting a score in HUMAB which says, I don't think this is human, so a negative score from HUMAB, there is a high chance that you have an antibody which will produce anti-drug antibodies. So it has learned about, if you like, the amount or potential for immunogenicity purely by being trained on this 1-0 classification problem. And I think this is really quite exciting because it suggests in some way the value of that score has some understanding of the, and I find it a very odd word, the humanness of an antibody, and can be used to think about how you can actually create antibodies which improve that score and therefore are much less likely to raise anti-drug antibodies. So the last thing I'm going to talk about today is something that we put very recently onto the bioRxiv. And this was really kind of built off the back of this improvement in structural modeling we have seen. So Paragraph was developed to predict paratopes. Now, we had tools from some time ago which were for paratope prediction, which had been overtaken by many other people's work. And what we wanted to do was try and think, given the techniques that are now available, can we do a very kind of minimalistic approach to this, put in as simple as possible features into a really good architecture? So we're going to use equivariant graph neural networks to do this, and see if we can just predict the paratopes of antibodies. So the input for us was the structure of the antibody, but actually we were just using the C alpha coordinates and only the C alpha coordinates of the CDR loops and 2 residues on either side. And the reason behind that is, though I am aware that there are paratope residues outside that space I've just described, if you use all of the residues of an antibody, you create a huge class imbalance. So you have very, very few paratope residues, and almost everything is not a paratope residue. And if you watch the training of machine learning techniques, when you have these very huge class imbalances, often, and we found this to be the case in our hands, they are very unstable. So sometimes they look like amazing predictors and other times not. And so it doesn't feel like a safe place to be working. Whereas if you just include the CDR loops plus 2 residues on either side, you barely miss any paratope residues, and about 30% of the things you put in are paratope residues. So it's just those say alpha coordinates, the amino acid type, and we, once again, I think Peter mentioned this, we found that using one-hot encoding rather than more complex language models as the descriptions of the residue types was the most powerful way to do this, and just the chain identifier. So was it the heavy chain or the light chain? We trained with both crystal and model structures because we always want to be able to make our predictions on model structures, because if you already had a crystal structure, you wouldn't be doing this. And what we output is the probability of each residue to be binding to the antigen. How well does it work? Well, the first thing I want you to look at on this kind of table of numbers is that top line, which is something we generated as a baseline. And really, I want you to look at this because actually I feel like I need to hammer this home in every talk I give. So those two measures, PRAUC and ROC-AUC, perfect performance would be 1. So in neither case do we have perfect performance for any method. That baseline method, all we did was we took what we used as the training data and we just predicted the proportion of residues found to bind at each sequence position in the training data set. So if every single antibody had a binding residue at CDRH3 position, whatever, we would say it was binding if it was 0.9 or whatever. So you can do that. What you can see is that is the best. Well, first thing you can see is actually on any metric, it's not very far behind what were considered to be some of the best methods for doing this in the literature not very long ago. And part of me feels like people really should think carefully about the baseline you are using to say, are we actually doing anything that is really learning something new and is actually predictive? And then the second thing is you can see that for ROC-AUC, it is actually the best performing method of all. And that is because ROC-AUC should not be used for unbalanced data sets. And because we have far fewer paratope residues than we have non-paratope residues, you can get very good results just by predicting nearly everything isn't a binding site. And of course, that inflates your errors, your kind of accuracy metrics. But now coming on to looking at paragraphs, what we can see is it appears to be performing very well, probably the best of the methods that are available publicly. And what is also nice in terms of the bottom line of the table is that it actually predicts better as we put more data in. So we expanded the data set. So in order to be fair comparison, we use the same data set that's been used for all the other methods in the previous rows of this table. But if you put more data in, it's kind of like I get improved performance for free. The other thing that we can do that improves performance for free is looking at the structures. So because AbLooper has this error estimate on it, I can say, well, how well would I do if I only use good models? And it turns out, as you can kind of see through the rows on this table, the better I think the model is, so this isn't how good the model actually is, this is just using my confidence estimate from AbLooper, the better my predictions are. So as antibody structure modeling improves, paratope prediction will improve for free. That's a very nice thought. And then the final comment on this is something that I think is worth contemplating on, which you can see actually, we start doing better on the models than we were on the crystal structures. And there are two possible reasons behind that. One is that as we start saying these models are very, very good indeed, it's quite a small data set. There may be some bias arising from that. Perhaps we are limiting it to things where it's easy to predict the paratope for some reason. The other possibility is, of course, we trained our method to predict on models. So actually, it's used to some amount of error in there. So at some point, these models are good models, but they're not crystal structures. Maybe actually the crystal structures are not the right things for us to be predicting on. Just to give you a picture of what paragraph prediction looks like when it comes out the far end, on the left-hand side is the actual paratope, the blue being the antibody, and you can see the red is the paratope, and the green being the antigen that is bound to it, and just two views of it, top down and side on. And then what we have on the right-hand side is the paratope prediction from paragraph. And there are two things that you can see from this. One, which we find really kind of quite exciting and we're starting to make more and more use of, is the fact that this, almost the higher paragraph score is, the more kind of central it appears to be to the paratope. And the second is the sort of slightly disappointing part of this, but obvious fact. Some of the paratope residues for this antigen are outside the region where we make predictions. So of course we don't predict them at all. You can see that some slightly red patch coming down. I don't know if you can see my mouse, but in the side on view, you can see there's a red patch coming down the side of the antibody on the picture on the far left, and we don't mirror that at all as we make no predictions there. So to sum up, I think one of the things I want to say, and I think it's a really kind of useful way of thinking about things from the way that we are developing tools, is adding structural information tends to make our data richer. If we can predict the structure well, why would we not give the model a hand by giving it that information as well? It is absolutely clear now that machine learning is playing a key role in computational antibody discovery pipelines. I think it's also clear that perhaps the biggest challenge is the lack of data. And I don't mean the lack of volume of data necessarily, I mean the lack of clean, curated data that can be input. A plea that I will make many times, and you will see it in many papers, is thinking and taking care about how we assess the predictability of these models. It is hard to do these tests well, to work out exactly how generalizable a model is, exactly how much better it is than what has gone before. And it is important that we really focus as a community in thinking about how we do that well. And I think fairly obviously, computationally designed biotherapeutics are on the way. It's just a question of when, not if. So just to finish, I should say thank you to my research group, most of whom are in that picture on the right-hand side. In particular to Brennan, Matt, Lewis, Alyssa, and Tobias, who did most of the work that I described in all of those slides. And as always, I should say thank you to all of the companies and councils across the bottom who give me money so I can actually do my absolute favorite thing and continue to do research. So thank you very much, and I'm really happy to take any questions you might have.
Thanks so much, Charlotte, for this absolutely fantastic talk, as usual. I have one question before we go to the Q&A questions. For the paragraphs, since antibodies can be cross-reactive, right? So they can bind, for example, two different antigens at once, and they might use different paratopes for that, maybe. I'm not so sure. How does paragraph would relate to that? Did you guys look into that?
So this is something which I find to be a really interesting question, and we don't necessarily have a perfect answer to. So in theory, and there's certainly a lot of evidence that you have non-specific antibodies or antibodies bind to multiple things. However, if you go and look in the PDB, there is not a single example of said behavior. So paragraph has never seen anything like that because it's been trained on the information we have about paratopes. I suspect, therefore, there are two effects. One, I suspect that potentially the paratopes are very similar across those types of antibodies, because that would make sense if that was the case. But as I say, we have no evidence of exactly what they are. And secondly, I suspect what would happen is, well, this is a big suspect. There are two possibilities. One, paragraph would become entirely useless if you gave it a very non-specific antibody, because it's never seen one, so it doesn't know what to do. Or its behavior would be an averaged behavior, because it would probably predict residues that are involved in both paratopes and then sort of score slightly lower for ones which are only in one or the other or something like that. But I would love to have data where we could start to explore that better, because I think it's a really interesting question. But it's one of the reasons why we didn't go for epitope-specific prediction, because at the moment, there is no evidence that we need to do so, given the data we have to work with.
Yeah, I see. So there is a related question for the paragraphs. So have you evaluated what proportion of the binding energy the paragraph predicted paratope residues contribute with for known antibody antigen complexes? Yeah.
We have looked at this a little bit. It's quite difficult to do systematically, because we can do it systematically if we predict the proportion of the binding energy, say, using a method, I don't know, like Rosetta or Foldex or something. And what you can see is something that looks quite friendly towards our results, which is that actually we are doing well at predicting the important residues. But I think, to be fair, I would rather do it with experimentally generated data, where there is much less of these single point mutations for us to work on, and very, very little where people have done an entire binding site and knocked them all out and given me all the numbers I need. There we still do see, but it's very small case studies. It looks like the higher scores relate more to more important binders, but I would hate to say that's actually completely true. Not enough data really to be able to do that.
Interesting. And then the last question also on the Q&A, playing devil's advocate, what do you think is the value of predicting thousands or millions of structures? Don't pharmaceutical companies just work on one lead at a time?
You see, I think you're starting in the wrong place. Just to be kind of straight back at you. When do you want to predict a structure? I don't want to predict a structure when I have one antibody that I think might work, because that's way too late. I want to know every property of every antibody that I am considering and discarding at every point along this. So if you are at the beginning of a campaign, even if you do a library search, you are going to have tens to hundreds of potential leads then. Then if you're thinking about what mutations you might make to those, you will have tens to hundreds of potential mutations. Instead of just randomly making them, model them all. Or I can go the other way and go, if I really want to understand the data in OAS, I need to look at what is the structural space of antibodies, not just the sequence space. So if I want to understand what is a feasible property for an antibody, I would say that the language models we currently have, and I think, I have to say, I really liked Conrad's approach of thinking about simpler language models as well. But those language models are starting to show, just like language models have shown in, general protein space, that you can have a description of that sequence space. I want to have a description of the structural space of antibodies. I don't think anyone's going to solve the million structures, so I'm probably going to have to model that as a beginning to be able to get a look at that picture.
That is also, that makes sense. So there's one last question, but I think we need to answer this in the chat. So Charlotte, if you could, so, and then we will move on to the panel discussion.